Assalamu alaikum. So this is the second portion of our lecture 2. In the first portion you learned how chromosome is created and the constituents of chromosome are DNA and a special types of protein which is histone protein. And there another protein was there, that is non-histone protein. So this was our last slide of the previous portion. Now let's repeat it again. Basically chromosome is composed of DNA and nuclear protein compact together in a supercoiled form, and these proteins are histone and non-histone. Histone proteins help in DNA coiling and forming its structure or giving its shape. The non-histone protein regulates the turning on or regulates whether a gene will express or not, and all of them are found in nucleus. This is a known information. Humans have 46 chromosomes, or more precisely, they have 23 pairs of chromosomes. Among them, 22 pairs are autosomes and one pair is sex chromosome, and that one pair will determine the gender of a certain organism or individual. What is chromatid? So far you have seen the picture of chromosome. Here you can see this is the whole chromosome, and this chromosome has this one arm and this is another arm. These are called the arm of chromosome, and these two particular arms are called chromatid. So the two exact copies of a chromosome that are connected together are called chromatid. The point where they are connected near the needle is called the centromere. Chromatids are made when new cells are going to be made. So far I know that all of you have learned the cell division systems, both mitosis and meiosis, and in that cell dividing stage chromatids are made. Making it clear, let's look at the picture. So this is the image of an original chromosome and this is a pictorial view. So this is one chromatid. This whole thing is one chromatid and this is another chromatid, and both of them are joined at the middle of their portion. It's something like a bow, creating a bow with ribbon. Both the ribbon are connected together at one knot at the center of the whole thing, and this center of the whole thing is called centromere. All the terminologies, chromatids, centromere, sister chromatids, all of them are very, very important. To have a clear concept about molecular biology or biological science or even genetic engineering, you have to know each term carefully and with their unique features so that you can have a clear differentiation among them. Like, what is sister chromatid? A sister chromatid is just like sisters. They're joined at one particular centromere and they are called sisters. And what is non-sister chromatid? Suppose there is a pair of chromosome or two chromosomes. This is one chromosome, this is another chromosome. And chromatid of one chromosome and chromatid of another chromosome is called non-sister chromatid. That means they are not sisters. They are non-sister and their chromatid is called non-sister chromatid. Please keep in mind this type of differentiation are very important. This is, as in the definition of chromatid, we have seen that two exact copies of chromosome are connected together. And here is the copies are formed. There is one single-stranded chromosome and this single-stranded chromosome is called chromatid. This is replicated, that means they form the photocopy of them and that are connected at the centromere. This is a double-stranded chromosome or the exact chromosome. And this is a picture of eukaryotic chromosome structure. Chromosomes are only visible when a cell is in dividing stage. So we usually see them in their double-stranded form. Basically, chromosomes are a micromolecular thing. That means it is hard to see them without any high resolution microscope. Moreover, they are only seen or they only become visible at the cell differentiated stage or the cell division stage, specifically at metaphase. And I think you all know what is the metaphase. It is the third stage of mitosis cell division. That is why what we always see the double-stranded shape of chromosome, and we call it the chromosome. And this one strand, this single-stranded stage is very short-termed, or this is the transitional stage. This cannot stay more than few microseconds. Within a few microseconds, they form the replication to form the exact structure of chromosome. Now let me make a clear conception about diploid and haploid. Both the words are known to you. In your intermediate level or in school level, you might have heard the two terms, diploid or haploid, or even if you do not have heard about it, I'll make it clear for you all. So what is a diploid? It is twice n. And what is haploid? This is haploid. So the HA is somehow omitted here, but this is haploid. This is the term. So what is diploid? Two sets of chromosomes found in all non-sex cells. That means diploid means there is two sets of chromosomes. And these two sets of chromosomes come from different parents. Look at the picture. This is one chromosome coming from mother, suppose, and this is another chromosome coming from father. And this set is called diploid. Diploid number of chromosome. Two sets of chromosome. So, alternatively, when there is only one set of chromosome coming from either parents, this is called haploid. Now let me give you the examples. In diploid chromosomes, we can see that one set is coming from either paternal source or maternal, and the other is from the alternative source. But the diploid formation or the diploid type of cells or chromosome pattern is found in all types of non-sex cells. Please keep in mind these are found in case of somatic cells or autosomes of an organism, with a few exceptions. For example, human have 46, crayfish has 200. That means human have 46 chromosomes, 23 coming from mother and rest of the 23 coming from father. And that is why they are called diploid set of chromosomes. If human have or any human cell have only 23 number of chromosome, that means it has a haploid number of chromosome. And these haploid number of chromosome is seen in germ cells, either sperm or egg cells. And I think you all know why germ cells have haploid number, because the total number of human cells should have 46 chromosomes or 23 pair of chromosomes. If one sperm cell has 46 chromosomes and egg cell have 46, so while forming a zygote or a new organism or offspring, this sperm and egg will fuse together, and this fusion will cause the number of chromosomes to become 92, which is totally a aneuploidy or totally a exception of normal calculation. That is why nature has made half of the total chromosome number in germ cells, so that while they are fusing together, these 23 number of chromosome is fused with 23 number of egg cells, forming the total 46 chromosome, which is our estimated number. Examples include human 23, crayfish 100. That means human germ cells. Here human means human autosomes. So I think you have now a clear vision about diploid and haploid, and please do not make confusion about their terminologies. 2n means 2n number of chromosome and n means 1n number of chromosome. This n denotes the sets. Now in the next portion, we'll see about some macromolecules and their basic features.